I think it's becoming very, very clear that the world is paying very dearly for not doing enough diagnostics early because, I think preventive and predictive healthcare is far more affordable than reactive healthcare. So fundamentally, if you want to think of the reimagination of health systems and the delivery of universal healthcare, this will be enabled by smarter and better diagnostics. And the more point of care, point of need that you can make it, the more effective and the more higher compliance that you will see.
Hello and welcome to another episode of Speciale Focus, brought to you by Speciale Invest, a leading early stage deep tech VC firm in India. I'm Hariharakli, your host. In this episode, I bring you a conversation with Dr. Vijay Chandru of the Indian Institute of Science, who spent decades navigating the journey from academic research to commercial applications. From co-founding Picopeta Simputers to pioneering Strand Life Sciences in genomics diagnostics to co-founding CrisprBits, Dr. Chandru's career tracks India's evolving ability to translate complex science into commercial products. He's also working on a new venture, Yantri Labs, developing a foundation model for financial markets inspired by Darwinian evolutionary principles. This one's currently in stealth mode, as they say in the world of startups. Strand's acquisition by the Reliance Group in 2021 was seen favourably by the capital markets. It's also an example of how deep tech ecosystems can develop through exits to strong commercial partners in order to achieve scale, Dr. Chandru says. The ability to scale does not always come naturally to innovators, as he points out. CrisprBits, now 3 years into development, applies CRISPR technology beyond gene editing to diagnostics and synthetic biology applications that could reshape healthcare delivery in resource-constrained settings. For example, CrisprBits has demonstrated diagnostic precision at 100% sensitivity for sickle cell detection on a variety of samples, including dried blood spots or cells taken from the placenta or from the amniotic fluid. The Sickle Detect test uses novel lyophilized kits that require no cold chain. The platform addresses an overlooked opportunity that most diagnostics globally rely on PCR or antigen tests, while CRISPR-based detection can deliver confirmatory molecular results with the simplicity of a do-it-yourself test. Near-term applications extend to food safety, livestock disease monitoring, and typhoid detection, areas where traditional culture-based methods impose significant supply chain costs. Beyond diagnostics, Dr. Chandru emphasizes a broader thesis: bioconvergence, the intersection of AI, molecular biology, and bioengineering, represents the next frontier for deep tech entrepreneurs in India. This conversation explores the technical constraints and economic logic underpinning molecular diagnostics at scale as India looks to develop its biotech sector to tap not only the domestic opportunity but global ones. So I was thinking about how to approach this conversation within the 30-45 minutes that I might have with you, and I realized you have done so much that the only way to do this is to focus on some very specific ideas and see if we can do justice to those ideas. So loosely, I thought I'll structure it by jumping straight into the story of CrisprBits first, which is what you're currently doing as a startup entrepreneur. And then as you go along, I thought I'll ask you about the ecosystem. And so let's start with CrisprBits. Tell us, in your own words, give us a big sort of snapshot of how that came about, and we'll go from there.
Right, sure. So, I basically was back in academia since 2017, roughly, right? 2018, I retired from all operational responsibilities at Strand. And so 2017, when I came back to IISc, I started, hanging out in the bioengineering group. Um, and essentially, I came back into sort of the research, uh, academic community in a more substantive way after a break of about 10 years. And when I did that, you know, started up a number of new project ideas. Um, I actually introduced courses in digital health and in computational epidemiology using agent-based modeling and other types of modeling systems. And that coursework that I initiated now has become a regular course at ISC. There's a momentum, um, and of course that became very prescient because the pandemic came. There were people trained to think about these, and ISC played some role in building, epidemiological models and predictive models, that were used during the pandemic by the COVID war room in the state and so on. Um, the other thread that I was looking at was with a group of classmates and contemporary uh, engineers from BITS Pilani. One of my colleagues, one of my classmates, actually a year junior to me, Rajiv Kohli, who's a professor of strategy at Columbia University in the business school, suggested that we look at CRISPR, and so we just started it up as a study group. We started reading, talking to some thought leaders. We had access to Siddharth Mukherjee and Sam Sternberg, Jennifer Doudna's student. And so we actually, I think, got quite excited. We also talked to some people in California who were, and then the pandemic sort of set in, and so we said, let's start something. But obviously, because of the pandemic, we had to go a little bit slow. We just basically set up a bench at CCamp and registered the company. It was actually exactly 5 years ago, um, in October of 2020, and then made a plan to start building out what we first called a workbench. So just to be able to order in uh, analytic samples, get hold of the technology and so on. But to why this interest in CRISPR was that it seemed to us that it was a fundamental tool for synthetic biology. And it seemed to us uh, that synthetic biology would play an enormous role going forward. I personally was excited because I had been very actively working with patients with rare genetic disorders since 2013, and of course the promise that CRISPR would actually be able to find cure for at least some of the significant Mendelian disorders was a very exciting idea for me. Uh, Others were looking at, uh, you know, the idea of smart proteins and being able to make good foods out of by engineering various cell lines and being able to grow food in a synthetic biology sense. Of course, people have made things like fake meat and so on out of these approaches. Um, there were also the possibility of getting into agricultural applications where you could actually tweak the genomes. And this is very different from GMO, right? This is actually doing a little bit of function enhancement without actually taking a whole gene from another organism and putting it into the plant, right? So in fact, by around that time, the Indian regulatory framework also was clarified that as long as you were doing small tweaks with techniques like CRISPR, uh, this would not be considered GMO. I think there's a threshold beyond which it's considered GMO. So you know, agriculture was another application, and and whole bunch of industrial applications because of being able to again engineer strains of microbes and so it just seems so fundamental that and we hadn't heard of any startup or industrial activity. There was obviously companies like Syngene and so on which do this as a CRO service, but we nothing that was dedicated to building platforms and product ideas, right? So that was what excited us, and so we were willing to put in some of the seed capital. So we sort of pulled in some monies, and uh, of course, first I would say almost till middle of 2022. First 2 years were kind of a little bit difficult, right? And the Omicron came, and of course, Delta was deadly. Then there was Omicron, and then so around I would say 3 years back, we really sort of settled into a rhythm, and very early on. Our former head of R&D from Strand was available. She had just stepped out of Strand, and so Vijay Gupta, came in, and she brought a couple of colleagues, and they sort of helped us build this first workbench. And then we demonstrated a diagnostic application. So it turns out that CRISPR can be used as a detection system as well. Feluda was popularized during the pandemic by the inventors at IGIB, and then later Tata's had picked it up, Tata MD. Um, I think the technology was improving very quickly, and we could see a lot of potential there. Um, and I think we actually experimented first uh, with actually surveillance of for SARS-CoV-2 and Omicron. We were actually able to develop a detection test for Omicron that was sensitive enough to be used on wastewater, right? And we then realized that in some ways this technology is, you know, possibly even more sensitive and more accurate than PCR. And so that was very exciting. So we said, okay, we'll push forward in the whole idea of being able to create diagnostics using CRISPR. And so PathCrisp is our platform for that. And Path, of course, short for pathology. So PathCrisp today is ready, and we've just rolled out our first product. It's still a lab developed test, LDT. It hasn't gone through the IVD certifications, but it's being run now in hospitals and for detecting sickle cell disease, carrier status, prenatal testing, and so on. So it's now, I'd say the platform is proven. We did a few clever things, of course, that will come out in press releases and so on. But we certainly published our work. We've patented it. And so the first, uh, product stream will come out now once since the platform is ready. We will make some, I think, significant progress on the clinical side. Of course, we're also looking at using this for detecting pathogens in livestock, detecting pathogens in food. Food safety is another, very unresolved area because I think traditional methods of culture-based testing for food safety and so on, um, is hurting the productivity of our food industry. So the business plan has been put together now, and happy to say that we're well on our way to closing. We went through seed, essentially with seed funding with our own founders pitching in, and one strategic investor called Vijay Bio, who are actually going to be soon manufacturing a lot of the reagents uh, needed for cell and gene research and industrial applications. And so we thought they found a good match. They said, you can be one of the uh, companies that uses some of these products. So they've come in as a seed investor, and so together with them, we have gone into a pre-series A round, because now that the products are ready, we need to fully commercialize and so on. And we're, yeah, in the middle, I would say towards the end of that pre-series A raise. We've raised about 80% of what we set out to. So the company is well poised. We moved out of C Camp early this year into the old Strand premises at the Veterinary Sciences College. UAS Alumni Association has a building, and we've taken the ground floor. It's about 7,000 square feet. Is just right for three clean rooms, and we'll be able to also do some manufacturing of kits and so on within that premises. We're now close to 20 employees and about 10 consultants, interns, and so on. So around 30 people. So I would say now we're a serious CRISPR uh, effort in the country, and I think will uh, complement a lot of work that is happening in the academic circuits. CRISPR is now part of any molecular biologist's sort of portfolio of techniques. And so there are a lot of people who use CRISPR, but we'll probably stay at a slightly specialized end of it. And so we're also doing a fair amount of editing services on making your usual knock-ins, knockouts, creating editing cell lines, editing microbes. So it's so all that is going on, right? And so we, like Stranded, we will have a services arm that is sort of short-term revenue generating, and while we build out the products and make that will eventually grow into the dominant business. But As far as actually going into clinical editing as a therapeutic, I think we're keeping that as sort of the third phase. So right now, the focus is really on the diagnostic products and the editing services. So editing services we call our EdiCrisp platform, and PathCrisp is our product stream for diagnostics. Yeah. So that's quickly the story of CrisprBits. Now, technically, I guess we are 5 years old, but I would say in the last 3 years we've been able to make a fair amount of progress. So,
I mean, even as a sort of general tech slash business reporter, and one gets to see that there seems to be a lot happening on the diagnostics front. I mean,
absolutely.
so in the big picture, can you give us a sense of CRISPR bits? You're going in a new direction. You're taking diagnostics in a new direction. I mean, what the
Yeah, I mean, yes and no. I mean, it's during the pandemic, of course, and even before that with Ebola and so on. Sherlock from the Broad Institute, Mammoth Biosciences from Jennifer Doudna's lab in Berkeley, all went down this path, and there were many others who followed in the West. In India, of course, as I was pointing out, Feluda and Ray, which came out of IGIB, showed some promise. Um, so, I think in the range of molecular diagnostics, right? We know that at one end of the range are there's PCR, right? Uh, and that, of course, became a household name during COVID with the RT-PCRs. Was sort of the standard of actually diagnosis, right? Uh, Now from RT-PCR, if you move up, you go to Sanger sequencing, right? Where you are not just looking at certain markers, but you're looking actually at you know um, a substantive part of the genome. And from Sanger, you go all the way to next gen sequencing. So this is the range of nucleic based diagnostic tests, right? You also have protein based testing. If you think of the rapid antigen tests, all of those. Now, the question is, can you get the ease of a rapid antigen test? What we saw as the Abbott COVID self tests, or what my lab was putting out, and so on. Those are antigen tests, but their accuracy was not great. Right? So even if you got a positive result, you would need to go and check it with RT-PCR, right? That was the standard, if you remember, during COVID. So the there's something about an accurate nucleic test, which is confirmatory, which doesn't happen with the antigen test. So we basically saw this opportunity to have confirmatory molecular tests with the ease of a self-test. Think of a pregnancy test, a COVID self-test. They're all protein-based tests, but now if you could get that same ease of testing with confirmatory nucleic signal, then you could say you've really arrived at not just point of care, but point of need testing, right? So I want to make that distinction because, I think point of care still means, a clinic of some kind, that has to be involved. You may have to set up PCR, and that requires, although they're getting better, more portable, and MolBio has done an extraordinary job at that. There is still infrastructure that's required, right? And so the question was, can you actually get there with CRISPR, which could, in some sense, be an attractive both. And PCR got to a point where you could get the cost per test down to very reasonable numbers, right? Uh, Maybe 500 to 1,000 rupees was what was available for COVID at high volumes. But so could you hit similar numbers, but at without the instrumentation and the or much lighter instrumentation? So that's where CRISPR fits in. Now the question is how instrument light are we? You know, where we will get there. But right now, what we need is we're able to manage with fairly crude sample extraction. Where we need isothermal amplification. There are two well-known methods of isothermal amplification. One is RPA, and the other is LAMP. LAMP is open. RPA went off patent just recently. All right. So standardizing on RPA is still going to take a little bit longer, but on LAMP we have it working. Right. With LAMP requires uh, the whole reaction to be run at roughly 55 to 60 degrees. So you need some heating device. Right. Uh, we actually have under development a very portable little battery operated heating device. But meanwhile, anywhere where there is a PCR, we can, instead of doing the thermocycling, you just heat it up and run the reaction. Now, so at least in wherever point of care was being done with PCR, we can go in with these kits and put them. The second thing is our kits are completely lyophilized. So they are freeze dried, and the entire master mix is in powder form and in microcentrifuge tubes, and so you just have to add sample and buffer and run the reaction. Right, no cold chain involved. Right, so this whole thing essentially travels by courier, whatever, and it's so uh, that's how we've been thinking about it. So at point of need, turnaround times which are you know under an hour ideally, but certainly under a couple of hours, right? Uh, and for things like you know food testing or pathogen testing, the next product we're working on is for typhoid. This is with ICMR, very close collaboration with ICMR. Not only typhoid, but typhoid resistance markers. So to actually know how to treat the typhoid. And these could be run, right at the bedside in clinics. You're not sending out and and so on. So that's the market segment that we believe is an exciting one. There will be obviously some situations and some uh, diagnostic applications where PCR will still maintain its position, but I think we'll be able to disrupt some of that, right? And so that's the thought process. I mean, and uh, just to give you a sense of this, we've just completed 130 clinical samples for sickle cell, and we have 100% sensitivity, right? So this has been done with hospitals and operated. The tests have been actually conducted by staff at the hospital and in our hands as well. And the tests are, precise. They're extremely precise. So the binding that happens in CRISPR between the CRISPR guide and the pathogen or the markers that we are looking for. The sickle cell is a germline mark. test. So this is actually going and finding that with great precision, so much so that we can call out sickle cell disease versus carrier. That is the heterozygous versus the homozygous cases, just from the CRISPR reactions, right? So it is, and just to give you a sort of, um, I would say, a more popular interpretation, the CRISPR guide finds a match, right? And then it sets off a Cas enzyme, which becomes like a Pac-Man. And the Pac-Man, can then start snipping nucleic material in the vicinity. And so if you throw in reporters with fluorescent tags, those get snipped, you will actually see the fluorescence. And in fact, in some cases, we're able to visually inspect this. So we believe that with a mobile phone, you should be able to take an image, get it diagnosed. And we built the whole cloud infrastructure to be able to actually read the data, analyze it, and send the report back. So it is. It's an exciting, kind of disruptive approach. We've been told in the food industry, for example, that when they make a batch of chocolate, for example, it has to sit for 3 days in the manufacturing facility until the cell culture tests come back. And that if we could actually run the tests in a few hours, then, that would be significant improvement in their supply chain. Right. So potential is high, and I think that's why investors have come forward. And just a note that all the investments that we're getting in are from non-VC funds. These are all family funds, strategic investors. So we've sort of stayed away from venture capital for now, because I think uh, they have a role later on when the commercialization is is in place, because then it's more predictable, right? And we don't get under those kinds of pressures. So I think the lesson there is also that for deep science, deep tech work, I think we need to find more patient capital. And as Sridhar Vembu also keeps saying, I think it took him 30 years because I think he wanted the time to be able to do things well. Right? So,
That's a really interesting point, and hold the thought on those lessons because I'm going to circle back to that. I just wanted to ask you, when you get to the point where you actually have a consumer grade product, like you said, it can just be shipped by courier. At that point, can you give us a simple explanation of how it might work? I mean, people will use what as the sample, saliva, or how does
right.
it work?
Yeah, I mean, so these things would vary, right? Because now you do need to get some sort of nucleic extraction, whether it is with, whatever there's a whole matrix of samples. It could be a dried blood spot, which you punch in, and so you make several punches in a dried blood spot, and then you heat it, right? And with that heat, it basically there is some nucleic extraction, and then run the reaction, right? It could be a cheek swab. It could be for things like sickle cell. You're only looking for germline, so it could be a cheek swab, right? Um, and so it depends on what you're testing. It's milk. If you're testing dairy, it could be a small bit of meat if you're doing it for the meat industry. For some of the livestock tests, like lumpy skin disease, which is a virus. You take a pinch of skin of the affected skin area from the cattle, and you can test that. So we've tested against a whole matrix of samples, and and of course, each has a slightly different extraction methodology, and you that's where the R&D goes in, and right, and accordingly it can be done. So I think eventually, can you make it all into a closed system? That is the goal, right? And are we there yet? I think probably a year away, right, from that. But meanwhile, uh, we've got the kits ready to do some tests. Yeah. Right. And the second level of complexity will be multiplexing. And right now we can do it with multiple tubes in a heat box, but that's kludging, right? You want to be able to do it with, say, microfluidic chip of some kind, and be able to do multiple readouts, right? So that is still pending in some ways, right? And it's all in R&D. So
Yeah, I'll come to that question in just a minute, but can you give us a sense of how far away you are from having a product that can ship by courier to a household that needs it?
right. So at this point, yeah, I mean, household, or say to a primary healthcare worker, a community health worker.
Yeah, or an SME, as you said, in
Yeah,
case of industries.
yeah, or these industries and so. So one product is shipping, that is the sickle cell, right? But it's shipping to labs, and uh, and I think, as I said, we're roughly about say 12 months away to actually get this into a closed system where it can actually go to households, and right, or be comfortably run by community health workers, right, in rural settings, and right. So
Can you try to give us a layperson sense of what you as a scientist would see as the holy grail of diagnostics?
see, I think, right. I think it's becoming very, very clear that uh, the world is paying very dearly for not doing enough diagnostics, and not and for not catching um, you know infections, or non-communicable diseases, markers early, because I think preventive and predictive healthcare is far more affordable than reactive healthcare, right? So fundamentally, if you want to think of uh, the future of the reimagination of health systems, and the delivery of universal healthcare, this will be enabled by smarter and better diagnostics, and the more point of care, point of need that you can make it, right? Um, you know, the more effective, and the more higher compliance that you will see, right? Um, so you know, it not everything needs to go to the house, right? Uh, and be done actually as a self test. A clinician can have a setup on his or her desk and be able to test the hormonal parameters, right? So, I mean, this is not what CRISPR is doing, but say what Achira is doing, right? Uh, and we're all in that mind space that we feel that the future of diagnostics is really going to have to be at that point of care, right? And of course, Strand, my first sort of experience with diagnostics is at the other end of the spectrum because it's really in lab, but it's very, it is meant for very, very sophisticated, testing. And there, it's it's important that, for quality control and so on, you do need to do it in a central location, and so samples have to flow uh, to the central location so that you can have high quality analysis. But that's that's also nucleic. That's also confirmatory. But that's it's the range that is needed, and I think all of these together will actually help us get to that goal of preventive and predictive and early detection, which is how you want your health system to operate. Um, I think there has been a misalignment in health systems for many, many years now. Uh, in fact, we had observed this when I was, elected as a pioneer, a technology pioneer at the World Economic Forum. I participated in the Future of Health, uh, you know, commissions and so on at the WEF, and we always talked about this misalignment in health systems where sort of the tertiary care and pharmaceutical industry, in some sense, their lobbies had sort of skewed health systems more towards reactive healthcare, and how to bring it back towards early detection, preventive care was the challenge because systems were misaligned. Okay? And I think COVID, I always say that, I think the silver lining of these pandemics, are always that something changes, right? I think the change that happened with the Spanish flu 100 years ago was a little over 100 years ago, was that was the birth of public health. Until then, people just thought of health care through eugenics, right? Eugenics was the underlying principle that, if you didn't survive it, it's because, you were not fit, right? It was only after Spanish flu hit the population, didn't matter whether you were rich, poor, what your ancestry was, what your it's that pandemic shook up the world, and it was the whole institution of public health started with the Spanish flu. Right? In fact, the Rockefeller Foundation was one of the first to help establish Johns Hopkins. They also came to India, helped establish AIMS. There's a long. It's a very important historical fact that many people have forgotten. The same way, I think COVID essentially has, you know, has to be a turning point, and technology driven, diagnostics driven, and AI driven health systems are going to be the result of that. It's I'm pretty convinced. It's not that technology's role in tertiary care and so on is any less. I mean, it still has a very, very important role there, and you can't. But I think the the shift in emphasis uh, was required, and I think that we did see. Right? Um, Now, I think this obviously also leads to questions about AI in health and so on. But I think it's all happening. It has to happen. I think it's to me, it's these are historical landmarks. That have to be. There has to be some pivot that happens. Mm. So
Mm. So, in the context of India's, need for population scale solutions,
absolutely, yeah,
in the field of diagnostics, you throw the story forward even 10 years, it's sort of exponential advancements in various technologies and sensors are improving big time and so on. Uh, what would that product or device or even solution look like, would you say? I mean, like a Star Trek kind of scenario, do you think that's not the correct way of imagining it, where almost like a universal diagnostics device where you can scan somebody and compare it to something on the cloud, and what are the problems involved there, because we are talking about even comparing somebody's genetic information to a database and things like that. So I'm sure you have thought about this very deeply, so I'm just wondering.
right, right. No, I have, and I actually chair the National One Health Mission subcommittee that looks at data sharing and and data integration. So you can imagine it's a hugely sensitive topic because you know um, you're looking at epidemics, pathogen outbreaks, effects of environment, of climate. So there are 14, now I think 16 ministries, departments, all of whose data has to be shared and with the right kind of compliances and restrictions on privacy, on security, not creating public scares and so on. And so restricted access and all of that. Um, so I think, let me just say that there are IT solutions that are possible. How well they'll be implemented. And this again goes back to our excitement about public digital infrastructure. And I think we we seem to now be almost world champions at PDI, right? I mean, you think about what we have done with Aadhaar, with COVID during the pandemic, how we were able to roll out 2 billion vaccinations and manage all the logistics of that. This was all done through public digital infrastructure. Were there a lot of lapses in security and privacy? Um, you know, you hear of some things, but was it that significant? I don't think so. I think India survived all this pretty well, and I think the same thing is true of uh, how we can handle a lot of these things. Uh, and I think things have become so much easier now with all your agentic wrappers around APIs and so on that can actually enforce various rules. And uh, so I think it has to be done carefully, with obviously with the right ethical standards. Um, but I don't completely buy that this cannot be done with a private public private partnership. You know, and frankly, some of that India stack infrastructure uh, is there in place to actually, and that is the public effort. And I think you'll have some primary healthcare handled through our government-run PHCs. You have a big chunk of second, secondary healthcare at district hospitals. But even there, we are starting to see that within district hospitals, the 10-bed ICU project, or coming in with it may be private philanthropy to begin with. I don't know if business models will evolve there, but you know, it. I think certain efficiencies come in when you do a mix of things. And if you look at how UPI was instituted, essentially as a mechanism for inclusive participation, but then how it also led to the whole fintech industry growth, I think. So private is involved, public is involved. It's all working together, right? And I think, can we pull off something like that for the health systems of India? I think that's the challenge for. Uh, unless we use technology in a significant way, right? I think it's going to be very hard to build a universal healthcare system with the kind of resources we have. I think on average, we need to be able to spend uh, at the minimum around 2,000 rupees per citizen per year. I mean, that's the number at which most states are trying to operate now, right? You have a range of Himachal or Kerala or some northeastern states which are spending more, and all the way down to 500 rupees per citizen in some of the states that are struggling with public health. But at some point, this is going to settle at some number, and within that budget, you'd have to roll out universal healthcare. It's going to have to rely a lot more on interventions at the primary healthcare level. It's going to have to rely a lot more on diagnostics that can give you early detection, and early detection makes all the difference, right? In terms of the cost of cure, or the health impact, health economic impact. So
In the context of what you've just explained, overall, what would you say are big opportunities for Indian entrepreneurs?
yeah, I think we certainly need a lot better sensors, devices, right? And yeah, you know, whether it's your watches, or various types of body sensors that there's you know brilliant opportunities, I think, across the board there. One of the academic projects that I'm doing is looking at the impact of climate and heat heat waves on health, and it's actually such an unexplored field, right? And we don't even know what happens when suddenly there's a 3 degree or 5 degrees higher temperatures for a sustained period of maybe a week, 10 days. What is the health effect, right? Um, unless you have good sensors and good ways of measuring, how would you even intervene, to be able to help people, particularly vulnerable populations and so on. So, you know, um, are these going to be wristwatches? Are they going to be patches? There's so many different ways in which this can be approached. I think mm, I think uh, there's an extraordinary opportunity on using biology and the convergence of AI, molecular biology, and bioengineering to come together. And now I would like to think of health in a slightly broader way, not just the human health, but one health and planetary health, right? Can we actually do something to fix uh, planetary health as well using this convergence of technologies? And so when the byline for our company, CrisprBits, actually, I'll read it out here. It's rewrite biology, right? Redefine health and reimagine the planet, right? And I think if you actually set out on these three goals, there's extraordinary opportunities for entrepreneurs. Um, and to reimagine the planet, I think I've been told by policy guys from the Hoover Institute and so on that uh, biomaterials need to be at about 40 to 50% of all manufactured goods. Um, we are at about 1/10 of that. It's only when we reach that 40 to 50% that sustainability of the planet is protected or ensured, right? So just being able to play a role in getting out of petrochemicals into biochemicals and and environmental cleanup using microbial methods, extraction of precious metals from waste, right? Using biological methods. I think it's a wide open space, right? And so the theme of bioconvergence is I think the the theme that entrepreneurs need to think. I mean, people have called it different things. I think um, Metzel has called it super convergence. I think Mustafa calls it the coming wave. So Susan Hockfield called it the age of living machines. So it's a very exciting time. I think people are very excited about AI, but I think it's actually the combination of AI, biology, and bioengineering and new materials. I think which is what uh, is the sort of it's that convergence which I think is the space that if I was a young entrepreneur, I would think seriously about. I'm not an expert to really tell you about execution in the field, and but there are some great role models, right? There, I think, our FMCG companies, our retail industry. There's obviously you have to bring all those elements together to actually have that impact. And in some ways, I think we have enough expertise in all of that as well, right? I think but I think it's this deep science, deep tech stuff that we have to get right. Okay? And if we don't get that right, you know, we'll continue to be in this situation where our IT industry, makes, per capita revenue of, let's say, around $50,000. And, the Microsofts make, $1 million out of each employee. I think unless we bridge those, unless we actually do the deep tech innovation here, and we're not going to see those values, right? And so I always say that this is the path to Viksit Bharat, right? That that we have to get our act together to make sure that we work on these deep tech innovations.
Fair point. Just to bring it back to CRISPR-Bits as we wrap up, what would you like CRISPR-Bits to be 5 years down the line or even 10 years down the line?
See, I think CRISPR-Bits is actually probably not going to be one behemoth company. I mean, I actually feel that since we are building platforms, there's probably potential to move this in several directions. But certainly, I hope one important direction, will be in bioindustrial applications, because I think that's a huge open space there, right? Um, One, of course, in diagnostics and healthcare, and with a diagnostics emphasis, and um, contributing towards universal healthcare in a significant way. A third would be actually going into innovation for therapeutics and using CRISPR-based editing. That I believe will come last. I mean, hopefully by the end of 5 years, we'll be seeing glimpses of that. But I think these two can happen right away. The Both the impact in diagnostics and health systems, and the bioindustrial applications of synthetic biology.
That's it for today. You can find links to Dr. Chandru's bio and CRISPR-Bits website in the show notes. And if you're an aspiring founder in India, especially in life sciences and biotech, do look them up. I'll be back with another episode of Speciale Focus soon. And until then, it's goodbye from me, Hari Arakli. Thank you for joining us.